attend the next seminars if I can, if you invite me to listen this time. So please put me on that email list, okay?
Allora, I will send you a link to the mailing list.
Oh, thank you. Thank you very much, Sophie. You're very kind. Excellent. So, as expected, I changed my title just because, you know, things are very dynamic at the microscale. So I would like to tell you about two things I have been doing since I moved to Trieste and I become a professor. Oh, mamma mia, I see all the friends from England, isn't it? Ciao. Excellent. So, it's nothing fancy. They are just two different approaches, how you can delve into the microscale, and you try to do the best with what you can do. Basically, that's my life. So today I will tell you something about marine, but not so pristine, and urbanized microbes. And we will go first in the top centimeters of sediment of the Venice Lagoon, and then we will go inside the ballast water. And we will discuss something about Pseudomonas aeruginosa isolate. Okay. All right, the roadmap. I just would like to advertise myself. So in case you would like to come to Italy, you know what I am doing. Then, of course, we all love Microscale. So I would like to take you to a tour at the Louvre. It's a special Louvre, you will see. We will then discuss the two study cases, so Meta-G microscale mining and ballast water, Pseudomonas aeruginosa isolates, and then we will conclude. Great. So
sorry, Francesca, just one thing, you're not in presenter mode.
Yeah, I know that. I can try. Usually Keynote doesn't like, or Zoom doesn't like it, but I can do presenter mode in case you stop me. Okay, if you don't see anything. Thank you, Roberto. Allora, okay, I hope I'm still there. Perfect, thank you. Allora, on the left side, you see the pandemic and all my students. I ran a lab with undergraduate bachelor students. So I kind of have a group of 5 so they can hand their hands, not to be scared, and do some great science. And mostly we are talking about pollution and pathogens that are human pathogens and arrive into the ocean. Then I have a previous life with OGS, and here you can see my collaborator, and today I will discuss the work I have done with Dr. Elisa Banchi in the sediment in the Venice Lagoon. And recently I have started interacting with the Pseudomonas aeruginosa project with the laboratory of Dr. Vittorio Venturi and his group, so Cristina Bez, Iris Bertani, and the student Cristina Chiara Maria, for understanding with a different perspective, genome-wise, the different Pseudomonas aeruginosa you can find in the marine environment. I am deeply attached to SIO and La Jolla. I am still appointed at CASPO, as just Jeffy is a real one. I'm just traveling there for fun. And I am part of a center that studies the influence of aerosol on the atmosphere and climate. Over there, I do marine microbial ecology in sea spray aerosol and how the biogeochemical cycles driven by microbes influence cloud formation, ice nuclei formation, and overall atmospheric chemistry. All right, let's think about Microscale. I know I'm preaching to the wrong crowd, but why not? So now I'm taking you to a virtual exploration, I would say, of the 3 best pictures that we have seen across our lives. I hope so. The first one I would like to show you is the molecular landscapes of a good cell, who is an exceptional scientist. He works at the TSRI in La Jolla, and he is an artist. So he's taking the high resolution electron microscopy images and then translate them into art. In here you see the very complex work of marine bacteria.
Francesca, we are still seeing the lab dimension slide.
Hey, I told you, thank you. I told you that sometimes it doesn't work, the screen sharing. So can I not be in presenter mode?
Yes, sure.
Okay, perfect. Allora, I need to somehow get out and maybe it's all stuck. Okay. All right. Can you see microscale thinking?
Yes, thank you.
Okay, thank you very much. No, I tell you that sometimes that's a weird thing to me. So I was saying, I was inviting you to a museum tour, and we are going to a special Louvre section where there are I think, the top 3 important paintings that are important when we think and study the microscale. This is the first one from Good Cell, Molecular Seascapes. This is a representation of the internal complexity of Escherichia coli. So Escherichia coli, as any other marine bacterium or any other bacterium or archaeum on this planet at the microscale, is very busy, interacting with molecules one at a time. And we are interested in understanding this intimate interaction because only by gaining this knowledge, we can understand fully ecology and predicting their role in the biogeochemical cycles. The second view, instead, is telling us how complicated is the microscale world. This is an impressionistic painting of my advisor, Dr. Farouk Azam. And the top part that you don't see because has been cut out from the science printing was his word where he stated the shapes of things to come. So with Roman, you explore the microscale world, and there is plenty of room at the bottom of things that we need to discover and measure and start thinking about that are structuring the interaction among bacteria, viruses, algae, bacteria, and also organic matter and bacteria. And the last one, of course, is Stoker, Seymour's Goric, that in this cube, very busy, where is life and death, are telling us that gradients are ephemeral and random, and they're heterogeneous in space and time, and you never know what is behind the corner. And the corner for marine bacteria usually is a blind corner. They cannot see, they only smell, and sometimes they move or sometimes they not. With this in mind, I like to place myself, I am a microscale girl, within the microbial loop. So the central part of any biogeochemical cycle of nutrient in the ocean is driven by the microscale action, the ecology and the interaction of bacteria and archaea with the organic matter. So let me tell you something, if we can summarize about the microscale thinking. Well, we know that the microbes interact with molecules constantly. Microbe-microbe interaction is an essential feature that we need to study and to address within any kind of ecosystem scale or investigation. Microbial action structure the ecosystem. They are the best environmental engineers. They provide ecosystem services to human by recycling organic matter, giving us oxygen, fixing CO2, and moving element. And they keep the ecosystem functioning, and also us, humans. So, to be kind of like Shakespeare will ask, or will pose, what is a microbe? What is the sense of being a microbe? We know that certain properties are shared by all the cells, such as metabolism, growth, and the fact that they are evolving, that means they mutate, and upon the contingency in which they live, mutations are selected and fixed over time. And then there are certain behaviors that are related to gene activation that are common only for some of them. Becoming a spore, having a resistant form, communicate with chemicals to coordinate gene expression, exchanges pieces of DNA in order to enrich their genome and chromosome, being able to motile, turn it on, turn it off, having flagellar, polar flagella or lateral flagella, according to the viscosity field. Produce secondary metabolites such as siderophore to acquire iron, or produce antibiotic to fight off the other competitors. And also change totally their lifestyle, deciding that they want to form a nice house with a polysaccharide matrix, some extracellular DNA, and some protein to give a sturdy, so biofilm formation. So they live in a very complicated world, and they never know what is happening. And the only thing microbes really need to is survive. How can they do it at the microscale? Well, they need to resist to antibiotic and produce some. They need to escape from predators. Viruses are always present, and grazers always ready to eat the right size of microbes. They need to explore the environment if they can, and the microenvironment, as we know, can have a different viscosity field, so they need to adapt their movement according to what they feel. They need to exploit nutrient resources in a very efficient way to fight off competitors. And then they need sometimes to kill others in many different ways, contact dependent, contact independent. And this is what, with these two study cases I'm going to present you, is just a view how you can, for instance, mine the meta-G diversity of data in a sediment community to ask a microscale question about what are they doing and how they are doing. With Elisa Banchi, a researcher at the OGS in Trieste, we were able to sample for 2 weeks, for 2 years, the sediment of the Venice Lagoon. We used grubs and sediment core, and then we extracted DNA and sent it to sequencing for amplicon and meta-G. We were interested in understanding who was there and which potential gene were they expressing. The idea is Venice, as you know, is sinking, and the Italian government has built submersible gates that also recently, maybe you have seen an image from Copernicus from the sky, that the bridge is up, and so Venice is not flooded because there was an exceptional high tide in the northern Adriatic Sea. Venice is also a very polluted area, and in order to manage it at the best, Italian government and scientists now know they need to manage the polluted sediments. Microbes, as you know, are moving nutrients and also metals and chemicals that are connected with the carbon cycles. So our first idea was to map who was there, and then which genes were present. And now we are still working on making a metagenome, assembled genome, and also checking at the transcriptome that was sampled at the same time in the same station during those years. What I can tell you is Venice lagoon is very big and heterogeneous, and we sample in 3 different basins: the north basin, Palude della Rosa, then the central basin where the city is, and the south basin where is Chioggia. Chioggia is characterized by sandy sediment. Instead, the central basin and the north basin is characterized by a mix of silt and clay. So what we saw was that we were able to see some fecal sewage, and so complex polysaccharide degraders and hydrocarbon degraders community in the top centimeters using the 16S amplicon, and we were targeting V4 and V5. So that was good. But we were interested more in the metagenomic data, since it was the first time people were sampling the Venice lagoon. Despite many, many studies I have addressed the chemistry and the biogeochemical cycles, DNA technology has not been exploited yet over there. So with an incredible amount of work, with 30 gigabase pair base, we had many, many sequences that have been curated, assembled, and then we subjected to seed and kegs. And so the take-home message that is not micro, but is macro scale, we see that the microbial community of the top centimeters in the Venice lagoon is structured by the site features, not necessarily by the season. And that was interesting to show because, you know, when the Venice lagoon is closed because the semi-submersible gates are up, the Adriatic Sea does not communicate anymore with the lagoon. So the majority of the metabolism is driven by this first top centimeter of bacteria and archaea. And so understanding that the structuring of this community is driven by the sediment type, the organic matter content, instead of the seasonality, was very interesting. Water column in the Venice lagoon can vary from 20 meters to 30 centimeters, and the water column, we know from previous studies, is more subjected to seasonal variation. The other take-home message is that we saw that the rare biosphere was supporting the microbial diversity within the sediment communities, opening up another black box about microbial interaction within the rare ecotypes present. We were then focusing on the microscale bacterium-bacterium interaction, and so we just cover the different mechanisms that bacteria can use to what? Exchange DNA, then protect themselves from viral attack, the system CRISPR-Cas, then talk to each other and also coordinate the gene expression and perceive chemical gradients. So what we saw is represented here in this bubble plot. If you see a star, it means that there is a difference that is statistically significant among the different sites. And so the take-home message is that Chioggia, that is in the center basin, is the sandy sediment type and is mostly influenced by the Adriatic Sea, is characterized by many motile bacteria that have a different chemotaxis pattern, maybe suggesting the fact that the sediment, the sandy sediment, has a larger pore size, and so the water column bacteria can permeate more in depth and still maintain their water column specific phenotype. Then for what regards the mobilome, that refers of all the genetic elements that microbe swaps, and they can carry important information in how to fight off antimicrobial compounds or how to resist to toxic metals, we see that they were very low in Chioggia, so in the southern basin, probably suggesting the fact that due to this highly dynamic microenvironment that the sand is in comparison to the clay and the silt, we don't have a lot of, there is no room for bacteria to exchange things. Possibly this is one explanation. Of course, we need to dig deeper, literally. Then in terms of cell signaling regulation and biofilm formation, we have a higher percentage of sequence were found in the southern and in the central basin that is characterized by the high polluted sites from the Marghera city. And that is interesting in terms of seeing the biofilm as a microbial citadel where bacteria and archaea needs to find shelter in this exopolysaccharides matrix in order maybe to decrease the total exposure of contaminant because of a decreased rate of diffusion within the polymer matrix. And then we saw anthropogenic related function, that means oil degraders or pharmaceutical degraders, virulence and disease or defense categories, that means toxin and antitoxin system, were very high in Marghera and Trese. So, of course, I haven't told you anything, it's just a shopping list, but these allow us to start creating hypotheses for when we will have the data that are in progress of the metagenome, assembled genome, and the transcriptome analysis that will be mapped on the MAGs to see which are the most active members of the communities, and given this microscale cell-cell interaction, which members of the community are using these strategies to survive. So in another way, with this meta-G mining, we are saying, we are asking the microbes to tell us how their microscale world looks. Now I will switch gear and I will tell you what I have been doing with my student. She graduated, Miss Francesca Iacozzo, within a project that was sponsored by the European Union. It was an Interreg project. BALMAS was focusing on understanding microbes, phytoplankton, zooplankton that are carried and they travel around our ocean in the ballast water in order to reduce this cross-contamination and the problems of invasive species. Lately, in the Northern Adriatic Sea, we have been pestered by actinophores that arrived in the Black Sea via ballast water, and it's called Memniopsis leidii, and it produces a lot of organic matter that marine bacteria degrades, but in turn, so it's good for the microscale part of the trophic web, but it's an avid predator and eats up all the larvae of the anchovies. So contaminated ballast water are a problem, especially in very narrow and small seas, such as is the Adriatic Sea. So we have sampled, you can see, the Gulf of Trieste just to see what was the signature of bacteria in a harbor area, so in a not so pristine urbanized place. And then on the right side, you see some flags that are telling you the ballast water that we have sampled in Trieste. Mostly they were coming in front of Zakynthos, the Greek island, and just one ballast water was sampled in Trieste, was loaded in Trieste and sampled in Ancona. So the ballast water was then brought into a microbiology laboratory, and we have cultivated only the strains of Pseudomonas aeruginosa. This work was done by my collaborator, Dr. Cinzia Fabbro of OGS. So we have 5 samples of Pseudomonas aeruginosa coming from ballast water, 7 samples coming from the harbor of Trieste, and 1 samples coming from the water column of the marine protected area in the northern Adriatic Sea. During this experiment, we have also investigated 2 human pathogens, Pseudomonas aeruginosa ATCC 27853 and Pseudomonas aeruginosa 01. In order to compare the behavior of this marine Pseudomonas aeruginosa and the strain. Of course, we have validated by 16S sequencing that the isolates were growing on media that were selecting for Pseudomonas aeruginosa, were actually Pseudomonas aeruginosa, so they are 99% or 100% with different 16S, but also different genes to the Pseudomonas aeruginosa strain that is a human pathogen. And so then we asked, thinking about the strategies that bacteria needs to express in order to be successful at the microscale, we then set up different assays. They will all run in Zobel medium, so in a very, very rich DOC concentration, 6 grams of carbon per liter, not 100 milligrams carbon per liter, and they were run at 21 degrees Celsius. All the isolates were motile, and so we check this during the growth phase, and we use simply dark field 10x microscopy. And then we ask the question, thinking about that the ballast tank is characterized by walls that are covered by microbial biofilms, and you are telling me that microbial biofilm is a very viscous word that is very heterogeneous, so maybe motility could be interesting to assay, but a different kind of motility, a swarming motility that use lateral flagella that are very long, the cells also become elongated. So we use a common assay, a 0.3% agar assay to test if the bacteria were able also to swarm. This is an image of the experiment. On the top 2 plates are a solid medium agar based at 1.5% that prevent the expression of the swarming motility. The top 2 plates are the same medium, Zobel, marine growth, at 0.3. And what you can see that over time, there is the bacteria change their expression, start producing hyperflagellate cells, and they start moving on like a raft. You can also see that there is some strange liquid going around. They also produce rhamnolipids that help them to reduce the surface tension, and so being more efficient in exploiting resources and arriving there first. We ran this experiment 3 times with the appropriate replicants, and we were able to distinguish 2 kinds of groups, the one that we called strong swarming and the swarming phenotype was defined in terms of measuring the diameter of the colony that was enlarging twice. And then we have the weak swarmers, and in the weak swarmers we have the two human pathogen, Pseudomonas aeruginosa 01 and Pseudomonas aeruginosa ATCC. Then we move on at the antagonistic assay, thinking about what Richard Long and Farouk Azam has done, and also Hans-Peter Grossart. We use the Bordorker-Agar diffusion assay, where we have create a lawn with a bacterium, and then we have spot onto this lawn 9 different isolates. So we did a 16 by 16 matrix with many replicates. And we were calling antagonism when we were seeing a clearance zone that you can see if you point your attention at the yellow arrow surrounding the overlaying colonies. So for fun, we also use a bacterium with what I have spent most of my life working, SWOT3, that is a Vibrio that was isolated by Richard Long during seawater bloom experiment. It was found in the attached phase, so on a particle. Is a strong antagonizer. It produces an andromide that is an antibiotic that inhibits the gyrase during DNA duplication, and it kills Vibrio cholerae. It also produces bioluminescence via quorum sensing, so it communicates. And we use it to see if how these Pseudomonas aeruginosa were comparing to this strong antagonizer that we know is SWOT3. And the answer was that, so we can, with this antagonism, we can define the percentage of inhibition capacity and the percentage of inhibiting isolates. So if you focus on the left side of the graph, what you can see is the number of isolates that a certain strain was able to inhibit. And if we look at the first one, that is SWOT3, SWOT3 was not able to inhibiting any kind of Pseudomonas aeruginosa marina, let's call them, that we have found. On the right side, in the bar graph, instead, you can read how many isolates were able to inhibit a specific strain. So SWOT3, despite the fact that it is a strong antagonizer when tested in the waters of La Jolla, of SIO, it was beaten up by 45% of all the isolates of Pseudomonas aeruginosa. The two pathogenic strains, PAU and PA at ATCC, instead were not inhibited by anybody, nor were able to inhibit. So something different is happening among the different Pseudomonas aeruginosa. And so then we can define, in this case, 3 different clusters of antagonism based on the Burg-Dorker assay. And the last thing, if you recall, it's important to produce some antibiotic in order to outcompete the others, and maybe also to produce some siderophores ligands that chelate iron, that is very limiting in the marine environment because it precipitates off since the pH is basic and not acidic. And iron is really important for any kind of respiration that microbes do. And so this is in terms of secondary metabolite production, it is known that Pseudomonas aeruginosa have colors, and colors of pigment are secondary metabolites. And now we know that some colors have another function. And here you see the combination of 2 colors that have been produced by these 16 isolates. Piocyanine and pioverdine. The first one is an antibiotic, is a redox sensitive molecules that it's when Pseudomonas aeruginosa in a patient that is suffering from cystic fibrosis is expressing this antibiotic that is a redox sensitive molecules, create oxidative stress in the macrophages and they burst. Pioverdine instead is an iron siderophore, and the Pseudomonas aeruginosa, again, that are pathogenic one, are using it to steal the iron from us. So, and in this way, depleting the host organism at their advantage. So we follow the growth of these 16 isolate for 78 hours, and then via a chloroform extraction, we measure the absorbance of piocyanine at 520 nanometers and pioverdine at 385. Pioverdine, being a siderophore, is pH dependent, so we were using the reduced form of the absorbance spectrum of this compound. And what you can see in the background, you see the OD, so the growth of this isolate, and we see an incredible production of piocyanine and pioverdine during the stationary phase, as we were expecting from the clinical isolates to do. And the diversity in terms of concentration of these molecules has a wide range, and so then there are different isolates that under this condition in Zobel, so totally happy, are producing a great amount of these two secondary metabolites. If we run some dendrograms, we can define three different groups or two groups: strong producer and weak producer of piocyanine and pioverdine, with this special isolate 25L being outsiders in terms of production of these two secondary metabolites. So far, with all the experiments that we have run, there was not a simple story that we were able to tell. So we didn't see any special trend in the grouping due to the ballast water versus harbour area. So ballast water and harbour area bacteria were scattered in terms of physiology and gene expression in the different groups. So if we take it all together and we look at the fitness being a multi-component phenotypes that we operationally defines in terms of being a strong antagonizer, being able to swarm, not being inhibited, be strong producer on secondary metabolites, and then being able to resist to some antibiotic, because my colleague in OGS previously has tested all these isolates for antibiotic sensitivities. We have three different groups. The red group on the right can be defined as bacteria, so Pseudomonas aeruginosa, that are mostly from the harbour area, with one from the ballast water. BW stands for ballast water, that have a behavior that prefers or support the production of a secondary metabolite and being a strong antagonizer. Then we have a group that is characterized mainly by being able to resist to some antibiotic. And then the third group is a mixture of being between medium and strong swarmer and being inhibited when the bacteria find each other in a non-contact way. The Pseudomonas aeruginosa, the pathogenic one, are always clustering apart. And then so we can say that despite Pseudomonas aeruginosa isolate are able to grow in marine conditions, the pathogenic one, they don't perform as well as these marine strains. Just hot from the press, we have finished the biofilm exploration. Of course, my biofilm is a more medical one. I'm not so fancy as you are, unfortunately. And so we run the common biofilm forming at in a 96 well plate at the air water interface. And what you can see is that the relative to the marine one that are clustered to the right side of this grouping, the two pathogenic one produce much less biofilm relative to the marine adapted one. And another thing that now we are trying to put together, my colleagues have been working on the same 16 isolates with another master student, Chiara Maria, and they have decided to sequence 7 genomes. And so here, what you can see, based on genome comparison, we have 3 different clusters that are matching pretty well with what we see. We have a cluster formed by 27, 17, and 22 that seems to be preferring a high production of secondary metabolite and being strong antagonizer. And then we have an intermediate cluster, 32 and 5H, that are transitioning from being strong to be intermediate antagonizer and strong to medium swarmer. And then we have a 6D, a C1F to be the least antagonizer one. So in the future, we are planning to do some more genome annotation and mining in order to understand which are the genes that the Pseudomonas aeruginosa, the clinical one, have lost in comparison to the marine one. And then we are going to do some antagonistic experiment with some strain that have been sequenced, the genome have been sequenced, that my colleague were able to put some GFP, some green fluorescence protein, against SWOT3 and other marine bacteria that are interesting in order to understand a more the fitness when they are living together, co-culture, and what is the importance of the secretion system type 3, type 4, and type 6 that are very important when they are fighting off either predators because they pierce the protists and the macrophages from the inside, the pathogenic strain, or when they are piercing each other, so killing each other using this secretion system. So this work would not be done without Francesca Iacuzzo and Dr. Elisa Banchi, the supporting agency Interreg, Balmas, and ICGEB, and Corilla 2020 Venice, the Venice project. Thank you. Ciao Eleonora, ciao Marco, adesso vi vedo.
Thank you, Francesca, for a nice talk. Now it's time for questions from the audience. Marco, I see your hand is raised.
Yeah, nice to see you again after so many years. So maybe I'll come and visit you in Trieste at a certain point.
Sì, please come.
So I was wondering microbiology you presented is over my my head.
Okay.
Way too much. But I was wondering how much does a community that is let's say captured into the ballast changes during the trip, that during the time it stays within the ballast, because the locations where the ballast water that you sampled were acquired close to Greece, right? Is they are at the beginning of the Adriatic Sea. Is, I mean, they don't seem to me to be dramatically far from Trieste, and yet it seems that by the time that they get to Trieste, then maybe they might cause a problem when the water is discharged into the Gulf.
Yes, so it's a very good question. So there are studies that have addressed the community shift in ballast water, and we know it's happening. It was not a part of the BALMAS project, so my colleague did not care about the bed bugs in terms of sequencing. But they were interested in seeing if the ballast water could be a potential way of seeding for pathogens, human pathogens that were coming from a long distance. So it's very skewed, and it's targeting the fact that do we see the same Pseudomonas aeruginosa in the harbour and in the ballast water that then will be discharged in the harbour? And they have different residence time, so it's very narrow. I'm not. It would be nice, I would say, to sample, the ballast water on the go, because they have a different resident time, and also the walls are not scraped. Usually, if they are lucky, they just put some hypochlorite, like when we go swimming in the swimming pool, this is what they do. And then they discharge it in the sea, like everything else. So the usual problem that we have with pollution. But the biofilm, we know, will resist the hypochloric treatment. So thinking about that, there are hitchhikers that are attached in the biofilm on the inside wall of the tank, and then they are discharged. Do I see a signature? But my work was more, the gist of it was, let's see what do we learn about human pathogens that travel the world, and they are still successful in being there. And so fighting off the marine microbes, and how do they fight off cell-cell interaction? So that...
Okay.
Ecco.
Thank you. Thank you.
Jonas.
Thank you, Francesca, for your talk. Very interesting. I have a general question. It's open-ended. In which environmental contexts do you think the cell-cell interactions are particularly important? And with the distinction between like this contact interactions or interactions mediated maybe by some external signaling? Yeah. Is there some consensus on this? Is this everything is everywhere in a way?
No, allora, the general rules, I would say, is viscosity matters. And so in which matrix you are. It's kind of the same stories we have with the enzymes. What is your strategy? Will you release an enzyme that stays in the matrix, and so it's a common good, but then at least you have something in return? Or would you keep it close to you? And so only you, if you have a good hydrolysis uptake system, can uptake it. That being said, so Eleonora was here in Trieste and I saw great things, but then, and, you know, from here and there, I always look, what is Roman doing? Allora, drang, 24,000 papers that I haven't read, and I started reading. So sediment and biofilm being, you know, high viscous and low diffusivity environment are good. Also phycosphere or marine snow. This is where you can have contact dependent and also contact independent. Then the rule is that certain bacteria such as Pseudomonas will pierce you only if you confront it. Others such as Vibrio cholera are always doing it. So also the gene activation that then I will say, I think we all agree, depends on the nutrient status, so depends on the microenvironment, can deeply influence how many ATPs do you put in building a nanoneedle to fight off. Ecco, so there is we know so many things about, of course, humans and the human ecosystem, that we are super rich, ocean always underexplored, and nobody cares about bacteria-bacteria interaction in general.
Thank you.
Eleonora?
Thanks a lot, Francesca. Super nice talk. Did you, I was wondering if you looked maybe more in details at the propensity of forming biofilm / biofilm composition in these isolates and how this could impact their survival. For example, so, composition, if they produce the same polysaccharides or not.
No, I didn't do any chemical things. I wanted to do a more outrageous thing that in simple terms will be co-culture, but in other terms will be using SWOT-free. That honestly, I was hoping this Vibrio that is terrible, is a pest, at least in La Jolla, and here in the Adriatic Sea, does nothing against the Pseudomonas marina. I was hoping to do some co-culturing experiment to see if there is a net increase or net decrease of the biofilm, and then try to find out who is doing it. Because you teach me that there is a lot of extracellular DNA and exotic polysaccharides that then maybe it's because one is overcoming the other one, so is killing. And so then I am using your cytosol, a pool, in order to make a strong biofilm. It's a little bit sloppy in terms of experiment. And so I will start first with the isolates doing contact-dependent killing, so co-culturing two strains that have a GFP, and then going to the EM and seeing who is killing who, by if I can have a high resolution of this secretion system, and also then seeing on selective media who is the winner. Possibly, I think you are right, there will be different matrices that this strain are able to produce, and but I didn't go there. I would like, of course, we can go together.
That would be great. Thanks a lot, Francesca.
More questions? I don't see any hands raised in the audience. So it's basically 6 p.m. So I think we can thank again Francesca. Timing was perfect. Thank you very much for your presentation, and thanks everyone for the very nice questions. So as I said, we'll send an email for the next meeting, the next seminar. It's going to be in January. We're not going to have a seminar in December. So yeah, stay tuned.
Allora, thank you, Roberto. Thank you, Sophie, and all the mob and all the friends that I see. And I think we need to say Merry Christmas and a Happy New Year. There is nothing else.
Oh, that's true. Yes.
Happy New Year.
Bye.
Bye.
Bye.